I'm molecular ecologist, which means that I use molecular DNA-based tools for studying mainly trophic interactions, but this could be applied to many other study questions or problems. And I have started my career by studying trophic interactions in rabbit beetles in French agricultural areas, which is very far from the charter focus. But through different opportunities and also very broad interests, I ended up being in the north and focusing on everything that's related to the tundra ecosystem, namely reindeer. And it was also quite, I'm actually quite glad that I joined Charter because it's the first time I will be part of such a big consortium. And working with great people that seem very knowledgeable about their study questions. So my first experience with, let's say, questions that are close to the Charter problematic started with my postdoc at the University of Oslo, where I was employed by the Rhinin project. It's a Norwegian Research Council funded project focusing on exploring very similar questions to Charter, actually see the impact of climate change and also the perception by local reindeer herders of how these changes are affecting them and the ecology of reindeer and other herbivorous species. So during the last 3.5 years, I have been mainly doing fieldwork, which was actually great. It was an incredible chance to get up in the north, collecting samples and then processing them in the lab. And now that we have all these terabytes of dietary data, I hope I can bring this to Charter and start working and analyzing them and drafting relevant questions that span some of the work packages within the project. Additional interest of mine, it's also all methodological challenges that emerge from using DNA-based tools. So I have been also involved in few studies that relate to testing different biases in estimating diet in reindeer with molecular tools. Like we did a reindeer feeding experiment. This was quite something, actually, to manipulate such large animals. And a few others. Working with reindeer herders, they provided us different gastrointestinal tract section of the reindeer so that we can compare and see. Regarding Charter and the next 9 months, I will be starting in March. I will be based in James Peat Group at the University Museum in Trondheim in Norway. And I will be contributing to 2 work packages, work package 2 and work package 4. Considering work package 2, which is more focusing on Arctic food webs, I think this is where my contribution from, with the Rhinin data would be relevant. So it's not entirely clarified down to the precise questions, but I think it will be very much looking variations in reindeer diet, especially regarding reindeer herding practices. We have sampled different districts in Norway where they have very intensive reindeer herding with large herd size, and also districts where it's more extensive, like less reindeer numbers, other aspects related to this as well. But this will be one of the focuses. The other one would be to track the changes in the proportion of lichens in the reindeer diet, as this is, lichens are something very central to these questions related to reindeer herding. Regarding work package 4, I think I will be contributing to this systematic mapping exercise, which is very new to me. I have never done this before. And I must admit that I'm less leading this part. I will be more contributing and waiting instructions. So I guess what would be relevant regarding my background would be to focus more on reviewing molecular data sets from the Arctic. Yeah, we have to say that DNA methods has been very successful in retrieving past biodiversity, especially in places when DNA it's very well preserved, like in the tundra ecosystem. So during the last 10 years, there have been several studies exploring changes in biodiversity over the past in the north. So I guess I can be relevant there in synthesizing these data sets. The easiest way, if you study diet, is to take the fecal samples because they are readily available and quite informative, actually. But when we started exploring a little bit how our perception of diet might change if we change the dietary sample, we actually realized that they don't necessarily say the same thing. So, of course, it's much less common that somebody would kill a reindeer, take its gastrointestinal tract, and use it to study diet. But there are several studies that have been using the rumen content, and not only in the north of, not only in reindeer or in general. So yeah, it doesn't necessarily reflect the same thing, especially regarding the temporal, how to say, coverage of diet. It seems that the rumen, or everything that's in the upper part of the tract, reflects a much more immediate consumption, let's say the last 24, 48 hours. While when you go towards the end, and especially the fecal samples, then we see that we accumulate, and potentially over a month sometimes, of diet. Yeah. First, yeah, even if you take a fecal sample and it's in the north and it's quite cold, we usually don't take it. We aim to get it freshly from the animals because, yeah, you still have degradation and etc. And you don't know in advance which plant taxa would degrade faster than other. So this has implications in the way you interpret diet. We're actually focusing on several different special templates. So what we did that we sampled monthly fecal samples from different reindeer herds across the Arctic. So in a way we can cover temporal variation at the month scale and also large spatial scales. We have samplings in Canada, in Yamal, Norway, and also we have few samples from Russian Karelia, which is a little bit more southern. But, yeah, of course, a lot of sampling in Svalbard, from Svalbard reindeer. But of course, when you start doing these types of, extensive sampling campaigns, you get into many additional opportunities. And it was really nice because in Svalbard, we had a master student that went up there, and she basically went almost every day during a period of 2 months observing reindeer. This was during springtime when actually vegetation changes very quickly, and collecting these fecal samples. So in order to see how with advancing of vegetation phenology and biomass and greenness, we can also track potential changes in diet. So we also have these samples. It's just the data, it's starting to accumulate, and we just have to move on with analyzing it. We have the chance to have a really good, what it's called, a computing server, computing capacities in the University of Oslo, where we store the data. Of course, we have several backups because they're not trust. But yeah, then once you receive it from the sequencing machine, then it's mainly bioinformatics to filter out the data. And then, of course, extensive steps of data cleaning, because yeah, people would be surprised, but we still lack common standards. Each data set, it's unique in a way. This is because of how the process of DNA analysis works. And then you have to evaluate where is the threshold of your noisy data versus your real data. And then you have to go back and forth and decide where you have minimized the noise while optimizing your real signal, knowing that sometimes you can also eliminate real data. But yeah, it's a bit an iterative process. You start looking the data, analyze do some, let's say, quick ecological visualization of data, then you go back, rechange some filtering thresholds, and then you decide, okay, that's my claim data set. I think it requires a lot of practice. It's definitely subjective, arbitrary, let's say, in certain aspects. But more you get experience with it, more you start seeing that, especially if you do some additional methodological work. For instance, we regularly sequence known mixtures of DNA, so we know exactly what must be there. So you start processing these samples with known composition, and then you start seeing, okay, what are the regular artifacts appearing? So then, in a way, you can get, quite good grasp after a certain amount of practice. How can I present this in a? It's sequences, and basically a sequence file, you can open it with a text file. Then what you would see, it's just lines of, sequence, let's say sequences, sequences. Of course, it's not recommended that you open a huge sequencing files with a text file. This would be like very long to open. But basically, yeah, it's just a series of sequences. Then, of course, you have to process afterwards because the format you get them from the sequencing machine, it's not readily available for ecological analysis. You need to convert into different formats, get rid of the large amount of, let's say, artifactual data, which makes the files very heavy. And at the end, it's literally, you can open it with Excel or text file, and it's a matrix of your, let's say, reindeer individuals and plants that have been found within their fecal samples. And of course, you can have the sequence associated with this plant. One very important precision, it's I never say that what we find in a dietary sample, it's what the animal ate. Because the presence of certain, let's say, taxonomic unit within this dietary sample can have different origin. So just say detected or retrieved or inferences about what is eaten, actually consumed and let's say assimilated are more difficult. Of course, the easiest way is to say that it's necessary eaten and assimilated because it's in the dietary sample. But because the technique is so sensitive, you can also detect things that might be, of course, contaminants. We try to minimize this. It could be also, you can have a DNA molecule for many reasons. But reading in papers that used to rely on this microhistological analysis, you get a piece of fecal sample, you put it on the microscope slides, and then you watch and try to identify based on the shape of the plant fragments. They mainly get to the very large groups of plants, let's say forbs or shrubs. Sometimes they might get the family, but this is much coarser than what we get. We can get genus and species level data, and almost all the time family level data.